It's a pleasure for me to present at the university, especially during these times. Also, yeah. So the topic of my presentation is designing functional surfaces for biological applications, from superhydrophobicity to high-throughput screenings. And as it was mentioned, my group is located at the Kарлsruhe Institute of Technology in Germany. And the goals of the research in my group are to develop functional materials, functional surfaces, interfaces, with the focus on biological applications. So we want to develop functional materials that we can use to solve biological problems, to answer biological questions. And what kind of questions we are aiming at. And here you can see some examples. So we are interested in understanding how we can use artificial surfaces to, for example, to control stem cell differentiation. How we can use surfaces or materials to control cell adhesion, proliferation, migration. How to control adhesion of bacteria and biofilms on surfaces, which is a big problem in medicine, in hospitals and industry. How we can deliver drugs, nucleic acids into the cell. How to accelerate drug discovery through the miniaturization of biology. And one of the topics, one of the aims of the research is also to kind of try to help to realize personalized medicine using using functional materials, functional surfaces. And kind of interesting, important question, how can we design or develop a functional material, functional polymer? And one of the important prerequisite there is, which is often forgotten by people, is actually interdisciplinary environment. And that's why I want to show our institute, which is, I think, is really important to be able to work in an interdisciplinary environment, to collaborate, to interact with biologists. And so my group is located at the Institute of, let me show you, at the Institute of Biological and Chemical Systems, Functional Molecular Systems. It's a new institute at the KIT, and it contains several groups, including chemists, material scientists, and pure biologists, and we're working in the same building, trying to solve problems of biological problems using materials and using chemistry. And another important requirement, of course, to develop any functional material, we have to control different things. We have to control surface chemistry, to develop methods to functionalize surfaces, materials. We have to control also physical properties, such as hydrophilicity, roughness, porosity, wettability, mechanical properties, and so on. Just a second. Then it's important to remember that biological interfaces, natural interfaces that are functional, they're not kind of homogeneous. They have patterns of functionalities, they have gradients of functionalities, and if we want to mimic some of the biological functions, we need also methods and tools to control both chemical and physical properties in space, to make patterns, to make gradients, to make different structures. And last but not least, also biological interfaces that are functional, tissues, plants, they are not static, they are dynamic. They change in time, they respond to stimuli, they release molecules, they have self-healing properties, self-cleaning, and so on. So this is what we are trying to do in the group. We're trying to first develop tools and methods to control these different properties, different levels, and then use them to create functional materials, functional surfaces. And let me just give you an example of showing the importance of surface physical properties for biological functionality, just to give an idea how important it could be. Here you can see a stem cell. This is an embryonic stem cell. It is sitting on a flat hydrophilic surface, and if we take the stem cells, put them on the surface, they spontaneously differentiate into different cell types. And this is a big problem because we want to use stem cells for regenerative medicine, where the idea is that we isolate stem cells from an organism, or we use induced pluripotent stem cells and then proliferate them in vitro to multiply the amount of the cells, and then differentiate them in a controlled way into a certain cell type, and then use in regenerative medicine. And in this case, once you isolate stem cells from its niche, it differentiates spontaneously usually. And some time ago, we showed that if you take the same cells and then put them on the same surface with the same chemistry, but with different micro-nanostructure, with this micro-nanotopography, these cells, they do not differentiate spontaneously. They keep their pluripotency for up to several weeks, and they proliferate in vitro on the surfaces. And then potentially can be used for regenerative medicine. So this shows the importance of the physical properties and not only chemistry, as many people think. Here's another example. If you make a hydrophobic porous material, solid surface, it has some functionality, but it's highly adhesive usually to microorganisms, to cells. But you can take this surface and then you impregnate it with a hydrophobic liquid, fluorinated liquid polymer, and through the combination of the porous hydrophobic structure and the hydrophobic liquid, you form a stable liquid layer on top of this surface, and this brings completely novel functionality. The surface becomes extremely slippery and repellent to different types of liquids. Has very interesting repellent properties, stability, and we showed that the surface can repel biofilms and bacteria, which was also demonstrated by Joanna Eisenberg from Harvard. She introduced in principle this surface and kind of revived it. But we demonstrated also that this substance, so-called slippery liquid infused surface, it can repel and inhibit adhesion of biofolders such as the cyprids, which are these are spores of algae and the spores of cyprids, which is a big problem in the industry, this biofouling of surfaces. But through this combination of porosity and liquid polymers, it's possible to inhibit this and reduce this biofouling problems. Let me show you some examples of surface functionalization methods that we have been working on, and we have some of the methods that we developed. And in my group, we focus on photochemistry, because photochemistry is, in my opinion, is one of the most convenient methods for surface functionalization, because it's a non-contact method, and also light is much easier to control spatially and temporally. So we can make patterns, or we can control irradiation in time, so we can trigger some effects, and also we can control energy of the irradiation. So I'll just go through some examples of methods that we have been actively working on. Work with photografting, thiol-ion and thiol-ene reactions. These are photopeak reactions, which are extremely one of the most simple reactions, but also very fast efficient. We use them a lot for surface patterning. We also introduced some novel methods, photochemical methods for surface functionalization. For example, UV-induced thiol-hole reaction for functionalization of surface hydroxy groups with UV light. We demonstrated that dopamine and phenolic compounds can be polymerized and the polymers can be deposited on surfaces by using UV light to accelerate this polymerization deposition, which is interesting because polydopamine coatings are substrate independent, and you can functionalize basically any material almost, including gold, Teflon, and all kinds of polymers. We showed tetrazolthiol reaction, which is interesting because the product of the reaction is fluorescent, while the tetrazole themselves, they are not non-fluorescent. It's a so-called self-reporting reaction. And we also showed UV-induced disulfide exchange reaction. I don't want to go into details for all of these reactions, but I want to show some of a couple of slides for this disulfide exchange reaction because it's interesting, because it's usually surface functionalization leads to kind of permanent modification and stable functionalization. But in some cases, for some applications, we want to have dynamic interfaces, dynamic surfaces that can be changed after modification, that can be reused or that are not permanent. And disulfide exchange is one of these reactions. It's known that disulfides under UV light, they can break into two thiol radicals, which then can recombine back to a disulfide. And if you take a substrate and functionalize it with a disulfide chemistry, and then irradiate this with UV light in the presence of a different disulfide, you break all the disulfide bonds, and then they recombine back into disulfides. But since you have an excess of a different disulfide, you basically exchange the this surface chemistry with a new disulfide. You change chemistry from hydrophilic to hydrophobic in this case. But what is interesting here is that you still produce a surface which is a disulfide functionalized surface. It's still the same chemistry, it's still photoreactive, and it can be then modified again with a different disulfide. You can take the surface and you can functionalize it then multiple times. You can switch back and forth using the same chemistry between different functional groups. And also this reaction is quite fast and can be finished within seconds. And since because of this dynamic nature, dynamic covalent bonds on the surface, we can think about new types of applications. For example, we can start from a surface which is non-fluorescent, functionalize it with fluorescent disulfides. Then we can take a photomask, a different non-fluorescent disulfide, and then we can kind of erase part of the surface, part of the fluorescent group, make a pattern, fluorescent, non-fluorescent groups. But at the same time, the whole surface again is covered with disulfides. It's completely reactive and can be then modified again with a new chemistry, or can be completely erased with using different disulfides. And here is another example of such an application of this dynamic surface functionalization. We can start from a hydrophilic surface and make a pattern, hydrophilic, hydrophobic pattern with hydrophobic disulfide. Then we can erase this pattern with a hydrophilic disulfide, go back to its hydrophobic surface, and so on. So we can kind of cycle this process multiple times. Now let me go to and let me show you some of the methods we use to control physical properties. And traditionally we work a lot with porous polymers. Porous polymers, because it's an interesting and simple method. And here you can see porous polymethacrylate, which was produced by polymerizing methacrylates with a cross-linker methacrylate in the presence of a porogen. Porogens are usually solvents or components that do not participate in the polymerization, and this leads to the formation of such a highly porous structure of interconnected polymer globules. And the important thing here is that through the composition of the polymerization mixture, through the nature of the porogens, we can fine-tune a lot of physical properties. We can change the size of the pores, we can change the size of the polymer globules, porosity, permeability, transparency, chemistry, obviously, and so on. And here you can see some examples of porous structures that were produced by a similar method, not only from our lab, different methods, but it shows the diversity of the structures that can be produced by this method. And the surface topography and structure and porosity is extremely important for the interactions with cells, with bacteria, with biology, because all natural interfaces, they are porous and they are rough. They are never just smooth and non-porous. And in my lab, we started for the first time applying this method to make thin porous polymer films on glass substrates. As you can see here, this is a cross-section of porous polymer. And we use all of the advantages of this method. We can change the size of the pores, we can change the size of the polymer globules, the thickness of the polymer, transparency, chemistry, and so on. And this brings me to another topic, which is superhydrophobicity. So if we take such a surface and produce it using hydrophobic monomers, for example, butyl methacrylate or even fluorinated methacrylates or acrylates, through the combination of this micro and nanostructure on the surface, the porosity, and the hydrophobic nature of the polymer, the surface becomes superhydrophobic. And superhydrophobicity, as you can see on these videos, it's an extreme water repellency. It appears because of the combination of the micro nanostructure porosity with the hydrophobic nature of the material. It is possible to make superhydrophobic surface using even hydrophilic materials, but that's not so easy. You need special geometry on the surface. In this case, these are amorphous materials. And in my group, we developed a number of methods to make superhydrophobic coats and surfaces, including such porous polymer films that can be made on different materials, which is usually difficult to apply to different surfaces. They have also very good stability because this is a three-dimensional material and not just a 2D material. We showed that cyanoacrylates, polymerization of hydrophobic cyanoacrylates can lead to such superhydrophobic coatings on different materials because cyanoacrylates, polycyanoacrylates, they are known to attach, to adhere to all kinds of materials. And you know, you used by yourself cyanoacrylates in a superglue. Superglue is based usually on butyl cyanoacrylate. And we showed that you can use this method to make porous superhydrophobic coatings on different substrates. We also showed that you can make superhydrophobic coatings using this silicon nanofilaments with the such nanofibers of silicon in diameter about 20-40 nanometers. And because of the small size of this silicon nanofilaments, they become not only superhydrophobic but also transparent. And because of the presence of the vinyl group, in this case, this surface is reactive. Again, it can be functionalized with thiols by thiol-in reaction. And this is interesting because in this way we combine superhydrophobicity with reactivity, and which is usually difficult because superhydrophobic surfaces usually require either alkyl groups or fluorinated alkyl groups on the surface, which are usually non-reactive. In this case, vinyl group is enough to make it superhydrophobic, but it's reactive, and we can make patterns down to sub-micrometer scale up to sub-millimeter scale. And this again brings me to the next topic, which is a kind of an idea to create novel functional materials through the combination of opposite properties, sometimes completely different. And in this case, we wanted to combine superhydrophobicity with extreme hydrophilicity or superhydrophilicity. So here you can see superhydrophobic surface, extreme water repellency, and this is a superhydrophilic surface, or extremely hydrophilic surface, which is produced by the same method but using hydrophilic chemistry. And both properties, superhydrophobicity and superhydrophilicity, they are interesting by themselves. They have a lot of interesting applications, potential applications, different also biological properties. But since we use photochemistry to make these surfaces, we can use a photomask and we can combine both properties, superhydrophobicity and superhydrophilicity, in the same surface in precise patterns. And this combination of these opposite functionalities, this leads to completely novel properties that are not present in the original material. And for example, if we use this photomask, we can create a hydrophilic micro pattern on a superhydrophobic surface, and this behaves like a microfluidic channel. You can feel this pattern with the liquid. You can even use these channels for separation of small molecules, like thin layer chromatography. If you use a different photomask, you can make such droplets of complex shapes that are confined by the superhydrophobic borders. So in this case, you can see 3 different droplets that are separated by a 100 micrometer superhydrophobic barrier. And this is interesting because the shapes of the droplets are quite unusual. And it's also interesting because then you can use this method to basically confine anything what you can add into water in this case. So you can add different chemicals, or you can add cells. And if you can add cells, they will precipitate on the surface. They adhere to the substrate, and you can make, in this case, 2D patterns of different cell types. So these are different cell types that are patterned through this method. And here is also interesting that to make a pattern on a surface with precision down to 100 micrometers or less, you just need the pipette, because the pattern is formed through the self-organization of water on a hydrophilic superhydrophobic pattern. So it's kind of convenient to make 2D patterns and using manual pipetting in this case. And such patterns of cells on the surface, they're important for fundamental biological studies, to study how different cell types communicate, how they interact with each other on surfaces or in tissues. So we use the same method then to make patterns of particles, or to make patterns of hydrogels, materials that are usually difficult to pattern using, for example, inkjet printing. And then when we use this method to make a microarray of superhydrophilic spots on a superhydrophobic background, we found that if you just then roll a droplet of water on the surface, you can in one step form thousands of nanoliter sized compartments in every single hydrophilic spot, and without multiple pipetting. So it's even single step multiplexing of one solution between thousands of compartments. And we can control the shape of the droplets, we can change the size of the droplets, we can position them very close to each other to make high density droplet microarrays. And then you can imagine that instead of water, you can use a suspension of particles, or solution of chemicals, or suspension of cells or bacteria, and then in one step form thousands of completely isolated microreactors and use them for individual reactions or experiments. And this is important, especially for biology, because in biology, high throughput screenings are extremely important. Pharmaceutical industries doing the screenings all the time for drug discovery. and they screen millions of compounds, literally, and the volumes that are generated in such screens are enormous. The costs are in millions of euros just for the screening. So over the last few years, we have been working on this kind of translation of this method, this observation, into a platform for biological miniaturized screenings, and we have shown applications of this droplet microarrays for culturing of cancer cells, primary cells, stem cells, for doing single cell screenings, to make also 3D cell culture, or bacterial screens, and so on. And 2 years ago, also started a spin-off company, Accuray, that is now trying to kind of translate further this technology into a product for screening applications, but also for personalized medicine, where screening of cells with patient samples is important, and where you don't have enough cells for doing the screenings. So from cells isolated from biopsies, just a few thousand cells, and you cannot use microtiter plates that are usually used for these experiments. So there's a kind of big problem there, and with this miniaturized platform, we can potentially solve this problem. So we are collaborating with some hospitals and research centers, pharma companies to investigate this, to try to make a product. Here are some examples of this culture of cells in droplet microarrays. In this case, you can see these are not cells, these are actually cell spheroids. Cell spheroid is a 3D organization of cells that were cultured, in this case, upside down. And in this case, cells do not have any surface to adhere to, so they start to attach to each other, and they form such cell colonies, cell spheroids, which are interesting because they, in such 3D organizations, cells behave more closer to the in vivo situation in comparison to the 2D cell culture. So pharma companies are trying to switch to this 3D cell culture because the results of the screening become more relevant and more kind of reproducible at the end. And here you can see another example where we used droplet microarray for to culture full embryos in each single droplet. So these are obviously not human embryos, these are fish embryos, and you can see that each single droplet contains a single embryo, and this is also important for screening of fish embryos because this is an animal model, and it's also kind of has advantages over 2D cell culture for toxicological studies. Another interesting, I think, example of application, here you can see hydrogel particles with a shape that was determined by the shape of the droplets. And in this case, we wanted to use droplets not to culture cells, but to polymerize droplets into a hydrogel in order to keep the shape. And then you can detach these hydrogel particles to make freestanding hydrogels with the shape defined by the droplets, by the surface patterns, basically. And now I'll go to the last project that I wanted to show today. And the idea of here was that we could use these droplet microarrays for biological experiments, for doing cell culture, but can we use these droplets for chemistry, or to use every single droplet to synthesize a different molecule and then apply it to biological screening? So this is the idea, would be to use the same platform to do high throughput chemistry on chip, then to use the same microarray for on chip characterization, and then make a library on chip, and then use the same platform, the same chip basically for biological screening. If we can do this, this would solve a big problem which we have now in in drug discovery field, where there's a huge gap between chemistry and biology, because chemistry is done as 100 years ago, basically in flasks and large volumes, and biology is much more advanced in terms of throughput, in terms of high throughput readout, analytics, and so on. And because of this, it takes a lot of time to create libraries of molecules and then to translate them into the biological microtiter plates. So if we could do chemistry and biology on the same chip, we could miniaturize not only biology but chemistry, go from milliliters to nanoliters. We can parallelize chemistry, do parallel reactions, combinatorial chemistry. And what is extremely important, we could integrate chemistry with biology on the same platform, which can potentially accelerate drug discovery and accelerate pharmaceutical improve the process of drug discovery. The problem is, if we want to use superhydrophobic, hydrophilic surfaces, the problem is that superhydrophobic surfaces are also oleophilic, because if you take an organic solvent with low surface tension, it usually masks completely the surface because of low surface tension, and therefore we cannot use superhydrophobic barriers to confine organic solvents, which are usually used for organic chemistry. So we had to modify this method, and we had to create not a superhydrophobic hydrophilic surface, but an omniphobic omniphilic surface. And this is possible if we get rid of all the roughness, porosity on the surface. In this case, to basically we want to get rid of the defects on the surface, and the defects, they lead to the pigment effect and to the spreading to the computer effects on the surface. So we use quite simple chemistry, functionalized glass or silicon surface with with a monolayer of such silanes containing vinyl groups. And again, because of the reactivity towards thiol-in reaction, this substrate can be then modified with different thiols, either fluorinated or hydrophilic, to make patterns. And if we use fluorinated thiol, then we found that indeed the surface becomes omniphobic, which you can see here. Even for low surface tension liquids, such as in hexane, the surface has very low sliding angle, which means that if you tilt this surface, this even low surface tension liquids, they can slide and be repelled from the surface very easily. So if we can make a pattern, hydrophilic or omniphilic, omniphobic pattern, then we indeed could confine even organic solvents such as ethanol and hexane into defined shapes, patterns on the surface. And here you can see some more examples of such droplets. And here you can see even hexane with very low surface tension can be confined in such surface. And this means that we can now use the substrates for doing organic chemistry using organic solvents, and then try to apply it to biology. And we designed a relatively simple combinatorial chemistry method to synthesize such molecules using amines, thylactone, and this pyridyl disulfide, which is a three-component reaction that leads to such a lipid-like molecule with hydrophobic tails, a linker, and a hydrophilic and positively charged head group. And these lipids, they self-assemble to liposomes in water. And because of the positively charged head groups, these liposomes, they can complex DNA or nucleic acids, RNA molecules to form so-called lipoplexes. And such lipoplexes, they can then deliver this nucleic acid into the cell. This is a process of cell transfection or gene delivery, which is important for biology, or also for gene therapy. And although there are transfection reagents, but not all are efficient, and there is a need for better transfection reagents. So we decided to make such a library of lipids using kind of sub-libraries of building blocks, thiolactones, and other building blocks to make a combinatorial library of different lipids with different functional groups, and then to screen them. But we wanted to synthesize them on chip, and then wanted to actually use the same platform for doing screening. So to do the synthesis, we took 2 droplet microarrays, one containing a mixture of thiolactone and disulfide in rows, and another with different amines. And then using sandwiching, you can kind of add amines to this thiolactones and disulfide to start the reaction in each single droplet. Then when the reaction is finished, you have a droplet microarray where each droplet contains a different lipid. What is also interesting here is that you can take this droplet microarray and you can kind of stamp it onto a multiplate, and then use this multiplate to do fast analysis by mass spectrometry of the synthesized lipids on chip, which is schematically shown here. In the next step, we take this library of synthesized lipids, and now here I just want to show you interesting way of, this is a proof of concept of parallel high throughput micro extraction using this droplet microarray. So you can also do potentially high throughput purification on the chip using this method. But we are working on some other methods, but this is interesting because this extraction is extremely fast in these droplets because of the small volume. And in the next step, we take this library of lipids and then sandwich it with a droplet microarray containing a buffer plus a plasmid DNA, so a nucleic acid. And this leads to the formation of not only liposomes, but also lipoplexes in each single droplet, based on different lipids that were synthesized in the first step. So we make basically a library of lipoplexes. Here you can see some characterization of the liposomes and lipoplexes. And in the next step, what we can do, we can take this library of lipoplexes and we basically print cells using a liquid dispenser into each single droplet to start the cell transfection experiment. We used plasmid DNA that is encoding green fluorescent protein, which we then use to monitor, to analyze cell transfection, the efficiency of cell transfection. The more cells become green fluorescent, the better the cell transfection efficiency. And here you can see some quantification of the transfection efficiency. This is a control experiment. And so basically we could find some lipids on chip that were efficient for cell transfection. But this was a proof of concept, but what is most interesting in this proof of concept is that we did on-chip synthesis of 75 lipids, on-chip characterization by multi-mass spectrometry, on-chip lipoplex formation, on-chip screening, and so on. And all of this, from the synthesis to the cell transfection experiment and to the identification of efficient transfection reagents, just took 3 days and 200 microliters of solvents, which shows the potential of this and the importance of miniaturization, parallelization, and integration of chemistry and biology. Because usually to do this synthesis of 75 lipids and then screening, it would take at least 3 months or more on one student. But because of this combination, miniaturization, everything becomes faster. Of course, it should work also. So with that, actually, at the end of my talk, I just want to summarize. In my group, we're trying to develop novel functional material systems. We develop methods to functionalized surfaces with UV light. We work a lot on superhydrophobicity and omniphobicity, different aspects of the special wettability. We work with porous polymers and hydrogels and liquid-infused interfaces. And then we make functional material systems, and then we trying to apply them to solve biological questions. So in my group, we have biologists working together with chemists and biologists. They have different biological questions. We collaborate with biologists working on stem cell research, biofield microbiologists, miniaturization of cell experiments, and so on. And with that, I'd like to thank my students group and collaborators funding, and thank you for your attention. I will be happy to answer questions in English or Russian or German.